Okay, so welcome back, everyone. Welcome to the second talk for today. So it's a pleasure to have Dmitry Rinberg, Dima. So Dima trained in physics. He got his PhD from the Weizmann Institute, then switched to neurobiology and discovered that there are dramatic differences between the awake and anesthetic states in the olfactory bulb dynamics in mice. So he was also a genius of farm and where he established the temporal limits to behavior in neural processing in sensing smell. Right now he's at the NYU Medical School, and so he will tell us start with the introduction to olfaction today. So Dima, the Zoom screen is yours.
Hello everyone, do you hear me well? Okay, so hello everyone, good evening, good morning, whatever time of the day you are. For me, it's 8:30 in the morning. I will be giving two lectures. Today we'll be talking about very general introduction to olfaction topic that I'll talk about beautiful system, olfactory system, what we know about it, where we stand. I'm not planning to cover all the grounds. I'm not I don't have any ambition to tell you everything, but I rather would like to introduce you to what subjects, to the subjects of the field, what's interesting, what's known, what's unknown, and where the field's is. And tomorrow we turn using olfactory system and actually deeply thinking about olfactory system, how to think about odor coding from the behavioral perspective. Because currently we can do psychophysics experiment, we can do behavioral experiment, but we can also perturb the neural system and interpretation of that, what that means, what the conclusion can we derive from that. It is still under development. It's not a subject that is well established, and I would like to talk about some concept in this area using olfaction of the system, and we'll talk tomorrow about that. But my talk is a little bit flexible. I will ask some questions. And honestly speaking, I did not time myself. So we'll see how far we'll go, because if we be very, very fast, I'll start tomorrow topic today. If we'll be slow, I'll continue introduction to olfaction tomorrow. So please raise your hand. Vijay will help me to navigate this. I prefer actually, if you raise a hand and ask a question, but if it's only by chat, then probably somebody will need to alert me because I wouldn't be following the chat. So, olfaction. First of all, olfaction is one only of the chemical senses that we have. There's other chemical senses in the animals, especially in mammals. Olfactory system, gustation. Interesting thing that gustation is organized. It seems to be solving similar problem, kind of sorting out different chemicals, but only in your mouth by tongue. But organizational gustatory system is very different. And from the computational perspective, it's very interesting why one system and another system is so different. But I won't be touching gustation. I will be talking mostly today, I will be talking about olfaction. And nociception. When you cut onion, you start crying. This is not olfaction. This is not gustation. You have a receptor in the nose. You have a receptor in the eyes that's sensitive to the perturbation, to the disturbance, and you kind of they like pain receptors, but it's also chemical sensitive. Actually, this sensation strongly contributes to your food perception. When you eat, or when it's part of your general perception, capsaicin, or all this perception is affecting your general food kind of perception. But again, we're talking today only about olfaction. When we're studying sensory system, what we need to talk about? We have multiple levels of understanding, and that's I wouldn't be the first who cited Mar three levels, but I do believe that that's kind of very nice framework to think about it. First, we need to think about computational theory, what the system is doing on a very high level. Why do you need olfaction? What signals is extracted? What the basic computational processing. Then we need to think about algorithm representation. So how specific stimuli are represented in the brain, how they process, and on and on and on. And the next level is hardware implementation. All these levels are definitely locked, so you cannot take one separately from another. But it's kind of crude schematics, and we'll be talking at different levels. And I would like you to pay attention at what level we're talking at each stage of the discussion. And what question should we ask? And there are three basic questions: how it works, how it's built, and how it evolved. And how it works, it's more or less computational algorithmic representation. How it is built, you do need to know what's actually organizational structure of the system and development, and how it comes to survival. And all this stuff, again, very much linked, because being a physicist, I like very much optimization principles. Or this thing need to be optimized for that, but you cannot evolve, there's a lot of other constraints that you need to think about it through the evolution, through the hardware implementation, and on and on and on. So this is all linked, and you need to think about it. And I will kind of alert about different aspects of our faculty system as we go further. What are we talking about when we're talking about chemical senses? We're talking what stimulus we need to do to have very good understanding of the stimulus. And to tell you the truth, that is a big problem in our function. Then from the stimulus to the receptors, neural network, and of course behavior. So unless you're talking about behavior, you don't know what the system is doing. You can do a lot on this level, on the receptor and network, but behavior is the final judge. Do you understand it? Do the system has been processed? Can you understand behavior? And definitely all these levels are linked, because behavior affects neuronal network and behavior affect receptors and stimulus. You probably already heard some lectures about chemotaxis. This is a classical example when we're dealing with a chemical stimulus and we study behavior and we try to understand how to optimize behavior to extract the stimulus to achieve the goal. But this is a great example when our function can, as a sensory system, can help us to understand high-level processing and high-level behaviors. What organism we're talking about? And surprisingly, in a system neuroscience course, I usually start with bacteria. Bacteria has all properties of the faculty system. It has receptors. It has not neural network, but protein network. It has behavior. It has adaptation. We can talk about dynamical range. We can talk about chemical recognition. On and on and on and on. But it's probably, not the only organism that we study. The next level is C. elegans, maybe fly larva, mouse, and of course human. I don't say that anything is simpler or more complicated. This is actually a very interesting question. What C. elegans, with all neuronal network that it has, can do what bacteria cannot do? What fly, with more elaborate neural network, can do what C. elegans cannot do? Is it better? What advantages do we gain as we're making the system more and more complicated? And then human, enjoy the wine and smell different aspects of the food. And why do we need? Why we didn't stop our evolution on the level, let's say, of the fly? Why the system of mammals are more complicated? Again, I just wanted to talk about, just to pull this question. I'm not going to answer this question. I just wanted to discuss. Usually at this moment, I like to talk about bacteria chemotaxis and talk about the behavior, but you have a lot of chemotactic lectures, my guess. So we'll go to a different subject. Okay, so why do we study? Sorry, So why do we study olfaction? What is the attraction to study olfaction? First of all, it's one of the most fascinating sensory system of humans. We have a few sensory systems, and olfaction is one of them. People suddenly became much more aware about olfaction due to COVID, when people start losing olfaction. Suddenly, I do have, and now I don't have olfaction, and it's actually affect my life significantly. So people start talking about it. But seriously, olfaction affect our life on the level that we don't understand, don't appreciate. And just for example, people who lose olfaction in adulthood has much higher chance to get to severe clinical depression than those who lose vision or audition. It affects our emotion, affects our some other processing, and we don't understand full biology of olfaction. But I will be talking more or less from information processing perspective. I will, again, my background, and I understand that many of you guys is from physics, computational science, and I would be talking about the aspect of olfaction, the pure biological, but I do want you to think about it, to know about it. Olfaction is a fantastic window to the brain. And it's actually relatively shallow sensory system, so very quickly processing get to the deeper brain areas, and you can do interesting behavioral experiment and try to understand the brain decision making, motivation, goal-directed behavior. You can use olfaction as your window to the brain, and it's kind of provide a playground to do basic experiments. We have a huge progress in vision due to the stimulus presentation, but to get to the cortex and vision, you need to cross 6 synapses. In olfaction, it's only 2. And I will show you at the end of the lecture tomorrow what nice, very simple experiment you can do to gain very deep understanding of the very basic principles that are, to my opinion, way beyond olfactory processing. And the last thing that we don't think too much from a neuroscience perspective, but that becoming very interesting topic. It's an interface with a poorly understood world of chemicals. Very interesting point that I don't know about other animals, but human body emit 1,800 different chemical molecules. From sweat, from urine, from saliva, from skin, blah, blah, blah. And they are around you, and they're surrounded. This is an amazing information field that we have no access. What's interesting that biology is taking care of that, and we not very much, even we don't take care about, or we don't use it very much consciously. We don't know about subconscious processing of this information. But animals in the wild use it all the time. Very simple stuff that animal in the wild can avoid another sick animal. Animal make a mating decision based on the smell of other animals. So this is an amazing informational field that is completely inaccessible. Humans have an amazing light sensors, have amazing microphones, have, can measure forces, can measure submicron movement, but we're very poor at measuring smells. We're very poor at measuring of all these chemicals. And we carry gas chromatographer with you all the time, which is much more sensitive than $500,000 equipment standing in the laboratory and doing one measurement in a half an hour. So we need to pay attention to that. We need to learn from biology how to deal with that, how to extract this information. So let's talk about the very basic stuff about what's the olfactory, why we study smell. And I would like to bring you the citation from Alexander Graham Bell, and let me read it. Did you ever try to measure smell? Can you tell whether one smell is just twice as strong as another? Can you answer the difference between one kind of smell and another? It is very obvious that we have very many different kinds of smell, and all the way from odor to violets and roses up to asafoetida. But until you can measure their likeliness and differences, you can have no science of odor. If you're ambitious to find a new science, measure a smell. And that is actually has been said in 1940. Unfortunately, we not very much further down the road in our ability to measure and quantify smell. And I will talk a little bit about it, but quantification of smell is a big problem. Google has a huge program of trying to predict smell from chemicals. We do a lot of machine learning, but there is no really theory. What is the basic problem here? So what's going on? Let's think what's happening in a color vision. In a color vision, you probably are well familiar with this color diagram, and where you have all colors that you can perceive. Along this line, this is monochromatic waves. And here you can basically you have a relationship between all the colors. Red is closer to the yellow, yellow is closer to the green, and on and on and on and on. So this is your perceptual space in relationship to the physical space. And if you have three sources of the color with this spectrum, that spectrum, and that spectrum, you can create any color inside of this triangle. And why that? Because we do know very well organization of the color system of our color vision. We know the receptor property. We have three receptors in our eyes. That is short, medium, and long wavelengths. They all have very overlapping spectra. And each individual, what's important, and each individual wavelengths activate multiple receptors simultaneously. Also, what's interesting, you cannot activate only green receptor without activating blue and red receptors. So it's always combinatorial coding, and there's no light unless you make a special tricks that is separate. That there's no light that can, let's say, create only green receptor activation. You never saw it. You always saw when you activate green receptor, activate red, and you a little bit activate the blue receptor. So we do know it very well, and we can predict the colors. If you have a spectrum, you can predict exactly the color that we will see. And it's also based on the fact that the lights are additive. Two photons hitting the retina, hitting the receptors, had an additive effect, and two colors are mixable, and on and on and on. And that has been discovered, and it was kind of studied way before we understood the receptors. And later we understood the receptors, and we know not only the receptor, we know the basic physics and optics of the eye, and we know the neural processing. If this is the road for studying olfaction, where we're standing, let's think about it. In olfaction, we have a bunch of chemicals. First of all, we have no idea how to quantify it. There's many attempts to put an axis in this field, and some are more successful, some are less successful. Because in vision, you have the wavelengths. You can characterize the wavelengths of the stimulation. You can create any arbitrary spectrum. Here, the spectrum is much more complex. You can create basically, there's approximately 10 million individual volatile molecules just from pure combinatorics, and you can mix them in any proportion. We know the receptors. In 1990, there was discovery of olfactory receptors, and we know the genetics of these receptors. We know the genes that humans have approximately 350 olfactory receptors, mice have 1200 olfactory receptors. This is genetically of all olfactory receptors. That's a lot, 1200. We know some of the biophysics or physics of the odor delivery. This is a nasal cavity of the mouse. Odor gets to the nose and touches the receptor through this very elaborate labyrinth in the nasal cavity. We know the neural network. We actually know a lot about neuronal organization of the olfactory system, and I will spend today a lot of time talking about this. And we can measure perception. We can present an odor and measure perception, but we cannot close this loop. We don't know how one odor what one odor evoke other perception, what perception evoke each odor. And when I say this, what do I mean by it? Let's say we have multiple chemicals, and it is not big secret. You can look it up, and the odor methanol smells like baked potato. So we do know that. And we know that methanethiol smells like rotten cabbage. And that chemical smells like toast. So we can create a table, and we can write all the machine learning. You can analyze a lot of data, and many of them probably super interesting machine learning. You can predict it, and but there is no really theory behind it. Like the theory of three-color vision, how to be perceived. There is no theoretical framework. There's again that predict from the chemicals to the perception. But there's another big problem. Let's take the three chemicals together, and it is almost impossible to predict that they create completely new odor object that is a potato chip. And that experiment has been done. If you create this chemical and ask, what do you smell? People don't call it baked potato. People don't call it rotten cabbage and no toast. They call it potato chip. So this is an object in your head, and we don't know what's going on. We know that the signal goes to the nose, and something happened in the brain that creates this object. This is a big problem in the field. How objects are formed, why three chemicals suddenly, in a very specific proportion, if you shift the proportion, suddenly it became, you do smell individual odors with the mixture of other odors. What's interesting, that is problem that is, I would say, complementary or orthogonal to different problems. Because basically, when you have a mixture of odors, you can train an animal, and actually you can train a human to understand individual components. So we can do the chemical detection in the mixture. This is a very difficult problem, and we don't understand how, in one case, you create an object, in another case, you can actually detect individual chemicals. And as an example of that humans are as good nose as other other animals is I bring always the perfumers. Perfumers have exactly the same noses. They have the same number of receptors. They've just been trained to use their noses differently, and they use the tricks, and they have been trained to identify individual chemicals in the mixture, and they almost recreate the formula. Not fully, they're still not a gas chromatographer. So these are problems that kind of interest me, interest the field, and we try to kind of understand from the very high level perspective. Okay, this was more or less introduction about olfaction, and let's go dig into the processing.
Ivan, is a question. You can.
Yes.
Okay, Arnab, can you unmute yourself?
Yes, sir. Can you hear me?
Yeah.
Yes.
Okay, so my question was that I myself personally have sensed it many times. I don't know for others. that sometimes some smells invoke the sensation of the taste of that particular substance. So is it related in the sensation pathway, or is it just an associative memory?
Well, how would you answer this question? Okay, imagine you have all tools in the world, just on the theoretical perspective. How you answer this question? What does this mean, evoking specific smell? I mean, let's forget the smell, it's too complicated. When you recognize a face, the information is on the retina. So if I give you a retina image, you can recognize my face, or computer can recognize the face. Or there is a cell deep in the brain that is a face recognition cell that triggers this. So what do you mean by, that's actually a super important question. And I don't know the answer. It's almost, what do you mean by that? Because information on the receptors, so if you give me receptors and I experience the smell, I can forget your brain and tell that that smell will be like that. Or I can go deep in the brain and read on the olfactory bulb in the cortex. Or maybe I train you to press the button every time you recognize the smell. So I will measure the muscle. So I don't know how to answer this question. So you understand what I mean by that? There's another raise hand.
Yes. Okay. Understand that.
There's another question by Sanket.
Yeah, hi. So I have this. I've been thinking about this for quite a while, like even before this lecture. So when we have sort of a visual stimuli, we can recall it. So we can probably hypothesize that there are certain neurons that store that information. But on the contrary, this is what I personally feel. If you, for example, someone asks you like, can you remember the smell of that particular object? So it is quite difficult. I mean, I don't personally think it's possible to recall a smell or an odor.
Yeah. So this is one of the psychologists like this comparison, that you can recall the image, but it's very hard to recall the smell. At the same time, you have a fantastic memory for smell, because I give you a smell and you immediately move to your childhood and smell, I don't know, the flower field near your house or whatever. We don't know. We don't know the mechanism. There's a lot of speculation. I'm actually a little bit, I don't want to go there, because there's also some kind of interesting point that I'm not definitely a specialist here, but people say that odor recall the smell odor recall the memory very easily. But if I show the image of the same field, you may, that smell that was evoked by the smell, you also will recall it. But maybe you first verbalize it. So there's a lot of cognitive processing that I definitely not an expert and don't want to talk about it. I don't know about it. But there's some, and there's, I would say, no hard science. So, so I don't know exactly how to, so what people mean by recalling the smell. I don't know.
Right. And I also think that this recalling the smell, I think it could be variable among people, like the ability to recall.
Yeah, absolutely. Very different. Very different. And again, perfumers remember all the library that's usually a few, I talk specifically to perfumers. They have library in their lab that creating the smell, about a few thousand smells. They do remember every one of them.
Yes. And
okay,
one more thing I wanted to add, like I personally feel like this olfactory remembering aspect, it is quite similar to what I feel is like the auditory stimuli, like about the amount of recalling auditory stimuli.
Maybe, but let's talk about it later. When I talk, I'm much more. I'm not would be talking about much memory, more initial processing. We have a few more questions. Let's move quickly because this is just introduction. I can imagine how many questions you'll have when I start talking about the substances. Okay. Anna Rosman.
Hi, sir.
Yep.
Sir, so smell, we get the sense of smell when the chemicals attach itself to olfactory cells, right?
Right.
So then can't we quantify the smell based on the proportion, the rate of diffusion and the amount of chemicals, the concentration of chemicals present?
Yeah, sure. But there's two approaches. First of all, really to do physical measurement, to measure individual chemicals independently, measure the mixture and have this quantification. The only problem that is the space of these chemicals is humongous. We can also project all the space to the receptor space and measure activity of individual receptors. And it's also not a much easier task because if you have 1,200 different receptors, you have 1,200 dimensional space. You're right, absolutely. So this is actually one of the directions of quantification of the smells using receptors, absolutely. But I will be talking about it later.
Okay. Okay, sir.
Yeah.
Quick question.
Yes, Professor. So I just the quote that you gave, gave from Alexander Graham Bell, right? That got me inquisitive. So I would just like to know where exactly are we in the science of measuring the smell? Are we have we taken some significant steps in that direction, or are we still at the origin?
Yes, we're taking, and actually there was we're taking significant steps, but you didn't solve the problem yet. That's what I'm trying to say.
So
So we're trying to measure smell, we're trying to predict this. There's a lot of computational stuff, a lot of machine learning people working on that. But as I said, we we don't have the unifying framework to understand that. That's my point.
Okay, Professor. Yeah, thank you.
Yeah. Let's move forward, guys. So, okay, hold on a second. I've been here already. I kind of move. Second. Okay, so let's talk about neural process. This is a cross-section of the mammalian brain. Actually, this is the mouse. I will be talking mostly about the mouse olfaction. Few disclaimers. I'm not by any way a species chauvinist. I think that all animals are worth attention. Flies olfaction is as interesting as mouse olfaction. Humans is a little bit harder to study, but humans can talk. This is a great advantage. There was a beautiful paper, humans as a model to study olfactory system by Noam Sobel. But I will be talking about mouse because it's a nice system. We have a genetics, we can do behavior. That's for me crucial stuff. And I will be talking much more about behavior later on the second lecture. And this system quite well studied. So this is a cross-section of the mouse nose. In order to sense the odor, mouse need to inhale it. The odor molecules need to get to the nasal epithelium and reach to the nasal cavity and reach epithelium. Olfactory epithelium, it's quite elaborate. It has a huge surface compared just to per volume. And then the signal goes to the olfactory bulb and then to the rest of the brain. So olfactory bulb in rat and mouse constitute approximately 5% of the animal brain, which means that in this animal, a lot of computational power, proportionally a large portion of the computational power dedicated to this sense. And this is only olfactory bulb. There's another cortex, another stuff. So mice and rats, rodents are very olfactory animals. Humans also, the size of the bulb is still large, but comparatively, it's much smaller than the rest. It's only 0.2%. But don't consider this is that we have a poor olfaction. Humans has actually amazing olfaction. We're just not always using, not always aware of that. We actually, it's hard to compare. Let's say for detection threshold, we're almost the same like for some chemicals like mice and dogs. Maybe chemical acuity not as precise as other animals, but that's some kind of interesting comparison. And mouse nose, just before we go to the brain, it's very elaborate. And humans and other animals has a very sophisticated structure of the nasal cavity. And this is just a picture of them. So fluid dynamics of this processing is very non-trivial. Okay, others get to the epithelium. What's happening on the epithelium? Epithelium has olfactory receptor neurons and the cell body located in the cell layer, and the receptors located in the cilia of the olfactory, this is cilia of the olfactory receptor on the going from the knob, and the cell body is somewhere here, but this is the structure of the cilia in the olfactory receptor neurons, and the receptor sitting in the cilia, and each receptor is the G-coupled sevenfold protein that has been discovered in 1990. The genetics of this has been discovered. It was a very critical discovery in the olfactory field. And different receptors sense different features of the molecules. And this is a very simple kind of idea, kind of lock and key, that the molecule sense different features different receptor sense have a different lock and key mechanism. And mouse have 1,200 different receptors. Humans have about 360 olfactory receptors. When odor attached to the receptors, they go to the through the epithelium to the mucus to the receptor, they activate the neuron, and when you have enough depolarization, the signals goes from the receptors down to the rest of the brain. What's going on there? As I said, receptors located in the epithelium, and each receptor is connected with the axons to the brain. And specifically, what is very interesting, and this is super important, that each receptor neuron expresses one and only one receptor gene. And all receptors that express the same receptor gene, let's say red, converts to the red glomeruli. And the blue converts to the blue glomeruli. And the cyan converts to cyan glomerulus, and so on and so forth. So this is a very interesting organization, nothing like this similar in the other sensory system. And that allows us to kind of channel information from multiple receptors to individual kind of glomeruli. We have mice have approximately 10 million receptor cells in the olfactory epithelium. And in the olfactory bulb, in the mouse, it is approximately 2,000 glomeruli in one bulb. We have two bulbs. So basically, almost each receptor has two glomeruli, and each glomerule has only one receptor. From the glomeruli, the signal goes to the next level mitral cells. And then from them goes to the cortex and the high brain areas. Crucial work has been done shortly after discovery of the receptors, when Peter Mammartz and others, actually it has been done in the same laboratory where the receptor has been discovered, in the Richard Axel laboratory, but Peter Mammartz and Van Vank and Catherine Dulac stained the receptor neurons with the same receptor gene, and they discover that they converge to one point, to one glomeruli. This is one of the very crucial discoveries of an organizational olfactory system. Now we take it for granted, but it was quite a breakthrough. So if on the right side you see the whole gene tree of the olfactory receptors, again 1,200 olfactory receptors, and if you mark one, you see a single or kind of glomerulus. You don't see another glomerulus because it's actually on the other side of the kind of, that's somewhere here. You don't see it here, but basically you can identify individual glomeruli in the olfactory bulb that is correspond to individual gene or individual receptors. What's interesting, and if you look at this tree, and this tree is basically built by the genetic distances between gene sequences, you can see that there is three groups. One big tree, another branch, and a third branch. And that's exactly correspond to three classes of receptors. Class 1 and class 2 that was discovered almost from the very beginning of when they have a gene sequence of the receptors. And that receptor is slightly different. I forgot the abbreviation. They're slightly different. The people don't even consider it as the receptors, but they actually behave in exactly the same. They don't consider it as olfactory receptors, but people always complain this is different type of receptors, but they behave in exactly the same way like receptors, and they create glomeruli, and they process the glomerulus in the same way like all other receptors. And what's interesting, the different classes of receptors located in different part of the olfactory bulb. And you see the red part that occupy the largest portion of the receptors is class 2. They occupy majority of the bulb. Class 1 mostly on the dorsal part of the bulb. Class 1 is here, and in between there is a TAR receptors. Nice thing about TAR receptors that they are very nicely positioned to image them. So that's actually a great advantage. And they all localize in the dorsal part of the bulb. Okay, let's go back to the neural network. So, as you remember, so what's going on? When an odor comes, it activates subset of the receptors, and different odors activate different subset of the receptors and different subset of the glomeruli. And we can open the skull here and put the genetic indicator in the receptor or in the glomeruli and image the activity. And I first talk about organizational olfactory system, then we'll talk about the processing, but let me give you a few words about the processing of the olfactory signals. So this is, you can observe this movie. Because when you present an odor, you can see activation of individual glomeruli, and you can quantify this. In this case, the butanol has been presented, and you can quantify the signals. This is the code. So somebody asked me, can we quantify odors? Yes, you show me the picture, I should have been able to predict what odor it is. The question is, if it is a mixture, if it's a concentration, that's becoming much more difficult. And again, we can create tables of this of the correspondence between odors and neural signature, but we don't have a theory behind it. And we can do multiple odors like that. We can create this, we can measure this, and we can try to understand the logic of this coding. And that's exactly what my lab and many other labs are doing, trying to understand the logic of this olfactory coding. That's going on the level of the receptors, but the signals goes from the receptors to the next mitral cells. And the glomerule itself is a huge, huge amplifier. It collects information from approximately 5,000 receptor cells and kind of process them, integrate, and sends to the mitral cells, and send to the next level, actually mitral and tufted cells, and which are processed further down the road. The organization of the olfactory bulb is quite complicated, and that is a relatively simple diagram what's going on here. So we have other receptors with the receptors located in the cilia of olfactory receptor neurons. Olfactory receptor neurons goes to the glomeruli. Glomeruli send information to the mitral and tufted cells. They're similar, but they're different. The mitral cells are located in the mitral cell layer. Tufted is a little bit more superficial, but there's a lot of granular cells in the olfactory bulb, which are interneuron, and all of them are inhibitory. And there's also periglomerular cells that are connected on the level of the glomeruli. And the number of type of the cells is large, but this is big classes that we used to talk about. Let's talk briefly about the organization of the disamplifier. You have 5,000 receptor cells converged to one glomerule. And kind of, it's a synapses, glutamate synapses, excited synapse. What's interesting that many receptors converge to a small number of mitral cells. So first of all, this organization is quite remarkable because it is common not only in mammals, but in insects too. It's a very similar organization. You'll be talking, you have the lecture from Kathy Nagel about fly system. I'm not going to talk about it, but I just wanted to mention, and I would reference to this a few times on my way, because processing of the olfactory information in mammals and insects are very similar in spite of many years of evolutionary distances. So something is very fundamental about this organization. You need to integrate information from multiple receptors to one channel. And I would like to pose an interesting problem and ask you to think about it. So there was a paper that presented in 2014. Actually, it was in the PLOS computational biology, so computational people should think about it. It's actually, so people measure receptors and analog of mitral cells. It's called PNs. In the insect system, it's also olfactory sensory neurons, but not mitral cells, but PN, principal projection neurons that goes from the olfactory lobe to the rest of the brain. And they presented an order, and they measured the receptors and PNs from the same glomerule. And now let's look on this very carefully. First of all, this is a timing of activation. And if you see for the concentration for 10 to the 4 in some units, the activation of the receptors come later than activation of the PNs. Also, if you notice that receptor threshold starts somewhere here or here, but PNs, you can see the signals even low. What's going on? How come PNs, that is the next stage of the processing, activated before OSNs? I actually would like you to think and answer this question. Raise the hand. Any ideas?
Dima, there's an answer in the chat.
Oh, in the chat. Sorry, let me look at the chat. Sorry, let me see the chat. Hold a second. Are the mitral cells similar to the mushroom body neurons in insects? Not mushroom body, in the olfactory lobe. Yes, they're kind of similar, but we need to take it. Well, periglomerular cell would help because they're getting signals after OSN anyway. So this is not really the situation. So first of all, I will tell you that there's no there's no breaking of physics law. Everything, causality is not. The problem with this situation is the following, with this experiment, that there's a lot of OSNs. Mechanosensation, very interesting, but anyway, in order to activate mechanosensation, you need to activate receptor first. And you cannot bypass the receptors. If there's another pass, then it should be order specific, because that was the signals order specific and concentration specific. It is just yes, this is a convergence, but how you can explain this? The problem with that, and you need to remember it, the air might not have other molecules. Then you wouldn't have any responses without other molecules. I'm just reading the chat. So convergence, this is a very important answer. Basically, what's happening, that the distribution of the responses of the olfactory receptors, let's say on a log C threshold is usually Gaussian. You have always variability. And when you measure arbitrary receptors, you try to sample it here. But you have one and very, very strong receptor that on the tip of the distribution. It's sitting somewhere here, but it happened to be a little bit more sensitive than others. And that's enough to activate the PN. So PN will be sensitive to this receptor. Okay? And that's why when you sample OSNs and average, you sample average receptors, which has the higher sensitivity, but PNs are so well tuned and sensitive to the first ever arriving signal and strongly amplified. What's interesting, you know, we have ability in our retina to detect a single photon, almost single photon. The same thing happens here. I don't want to claim, and it's never been measured, how many molecules can we sense. But we can measure part per trillions, I forgot all these numbers, 10 minus 14, 10 minus 13 mole, the very low concentration, and you have a humongous amplifier here. Basically, this all glomerular unit is amazing, a very powerful amplifier that amplifies any coming signal. And that's why we see this very interesting effect. Now, what's going on further? That cannot be functioning all the time like that. And you know, you're not always sitting in the dark and recording individual photons with your retina. Retina need to function on the daylight. The same thing with the receptor, with the nose. You bombarded with many molecules and you need to process. There's a lot of other cells that contribute to the processing. And there's two types of the cell. First of all, periglomerular cells that are taking synapse from the OSNs here. Let me use a different color here. From OSNs here and synapse to different to other glomeruli. And there's a bunch of granular cells. And both of them have excitatory, so no, sorry, not that. So the synapse between mitral cells and granular cells are very, very interesting. This is, it's excitatory towards, it's excite granular cells, and granular cells inhibit mitral cells. So granular cells are inhibitory neurons, but this is so-called dual synapse, or binary synapse that has both excitatory and inhibitory in opposite direction. And I would like to make a deviation from here and tell you an interesting story. And when I think about computational neuroscience, I often cite this story because this is quite amazing. If any of you, I know that many of you was coming from physics and computational background. If any of you dealt with the astronomy, do you know the discovery of the Neptune that happens 100 and almost more than 150 years ago? And in September 24th, 1846, you can imagine computational capability at that time. And the Neptune has been discovered in a very interesting way. Verrier did mathematical analysis of the deviation of the Uranus and predicted that Neptune should be in a specific area of the sun. And Johann Gottfried Galle with the Hendrik Louis Diarist put the telescope and found it on that day. Very precise position of the telescope that we were able to find the depth. So it was a manifestation of the computational astronomy, how the planet was found. Well, how that's related to our story? In 1966, 1966, 50 years ago, Winfried Rao, a very famous computational neuroscientist, was working with Gordon Shepherd. Winfried Rao passed away a couple of years ago. Gordon Shepherd is still professor at Yale, and he did a lot of fundamental work in olfactory processing. And they did the following experiment. They put an electrode in an olfactory bulb and moved electrode through the different layers, from the glomerular down, 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 and they stimulated the nerve that goes to the brain and measured the responses of local field potential. And what they observed, that if the electrode is very superficial, the response like that, at some moment, it changes the polarity. And Rao, so Shepherd was measuring this, and Rao was modeling this. And Rao predicted that the position of the excitatory and inhibitory synapse, there was an idea that signals goes to the mitral cells that excite granule cells, and granule cells inhibit mitral cells. Rao predicted that the position of the synapse should be very close to each other, inhibitory, excitatory. They call Rees, and they put it in the microscope, 2-photon microscope, and they found the synapse that has dual side. This is one direction. You can see the synaptic vesicles on one side, and there's another. That was a computational discovery. They put a telescope and directed on the right part of the sky, and found the proper synapse. I think it's an amazing discovery. Not many people know about it. That's how the synapse has been discovered computationally, and then by EM in 1966. So it's quite inspiring for computational people. We're trying to solve different problems now, but anyway, this is remarkable. This is dual synapse. It's called dendrodendritic synapse between mitral and tufted cells. The big question, what is the role of that? What the computational role of that? And the last 50 years, we tried to solve it, and we still don't understand it properly. We have some hypothesis of computational role of that, but we do need to know how it functions. Any question on this part?
Dima, there's a question on asking what properties of the mitral cells leads to signal amplification.
Very good point. It's not only property of the mitral cells, it's actually property of the whole glomeruli. So basically, and I'm not going to discuss it in much more details, but we have here glutamate synapse, but also we have integration of the signal from multiple receptors. And there's an interesting phenomenon of the positive feedback of the glutamate spillover when one synapse, one receptor sends so much, one synapse delivers so much glutamate that nearby synapse also starts sensing it. So on this level, there is a lot of integration and amplification happening here. On the level of mitral cells, it's the signal from the dendrite going to the mitral cell body, and then the spike generated here, but that's general cellular biology. But amplification happening in the main amplification happening in the glomeruli. More questions? Nope, sorry.
So far.
I try to move forward through the slides. 1 second. Okay. Okay, now there's interaction between glomeruli, and interaction between glomeruli happens on 2 levels. First of all, through the dendrodendritic synapse and granule cells, and second, through the PG and so-called short axon cell that got excited from one glomerulus and inhibiting other glomeruli. And that diagram is kind of also very interesting and very general, because it's actually very similar to the retina. look at this organization. I don't know how many people are familiar now with the retinal organization. We have photoreceptors. We have one level across inhibition across photoreceptors. We have ganglion cells that sends information to the rest of the brain. And what is here that is the specific synapses are different, but we have two levels of inhibitory networks. First layer on the level of glomeruli, and second layer on the level of granular cells, or ganglion cells, or mitral cells in our olfactory bulb. It's quite a remarkable diagram, to my opinion. Actually, I do recommend this book. It's written by the Shepherd, the same Shepherd who was on the 1966 paper. And I will, sorry, I don't have the full reference. I will send the, if you ask me, it's a synaptic organization of the brain. This is a picture from this book. This is a more modern diagram of the whole bowel processing. You have glomeruli, you have PG cells, you have short axon cells that are actually project the axon far away. Some of the axons goes as far as 30, 40 glomeruli. You have mitral and tufted cells, you have granular cells, you have internal so-called Blaine cells that talks between mitral and granular cells. There are many kind of a lot of different cell types. What's interesting that we forget that there's approximately 50,000 mitral cells, and granular cells is about 5 × 10⁶, 100 times more. And why that? We don't know. Why we need so many interneurons, what their function, we really don't know. But there's some theory about that, and I'm not sure that we'll have time to cover all this stuff now. Again, historical review. One of the initial schematics of the olfactory system has been done by Ramanathan in 1911. This is a beautiful picture of the glomeruli, receptors, mitral and tufted cells and mitral cells that project their signals to the cortex. This is a cortical processing. What is amazing that already Cahal found the neurons that send signals back. You see this arrow? And telling that the cortex talks to the bulb. This is great prediction based on anatomy that some signals goes back to the and specifically connects to the granular cells. What Cahal found, because this is the granular cell that connect to the mitral and tufted cells. It is 1911. We have a lot of speculation why that, or what the purpose of this feedback, but there's no computational theory about this. Okay, so what's going on further? If we we talked about bowel processing, what's going on after the bowel? Mitral cells send information to the cortex. Where it going? So in 2011, we 100 years after Cahal, in the work of the Bob Data laboratory, again the same name, Richard Axel, and the first author, Darasa Sasulsky, were able to mark all mitral cells connected to the same glomerulus and see where they project. And what they found, that they project everywhere. They project to the many portion of the mouse brain. This is anterior olfactory nucleus. This is actually lateral olfactory tract, LOT. They project a big portion project to piriform cortex, entorhinal cortex, to amygdala, and olfactory tuberculate. And they create a whole massive projection and how to make sense of it. What they did next, they compare projection of one glomerule to projection of another glomerule. And they applied many statistical analyses trying to understand if that glomerulus is somehow different from that glomerulus in their projection. That is based on the idea that different part of the retina projected has a very specific mapping in the cortex. Even through LGN, we still preserve this mapping. Can we find the distances? Can we find the projection? Can we find how different receptors are projected? And they didn't find anything. Basically, all they attempt to find the structure here, can we discriminate statistically between different glomeruli or any random glomeruli, kind of failed, which led to the ideas that projection from the olfactory bulb to the cortex is random. What that actually entitled? Why this is cool and why it's not cool and what's actually going on? And by the way, when they studied the projection in amygdala, they found that different glomeruli actually project to different parts of the amygdala. You can see the differences in the projection from different glomeruli, which leads to the idea that the cortex is arbitrary learning machine, and you basically have a lot of random projections, and you don't have a specific map in the cortex. You don't have a specific map in the cortex, and but amygdala, who is more responsible for innate behaviors, which is analogous, there has been a lot of studies on flies. So some smells evoke attraction, some smells evoke aversion. That is more innate behavior that is processing in amygdala, and the cortex is a general learning machine. Random projection is a cool idea because you immediately bring the already multidimensional space of the receptor activity to even larger dimensionality by random projection, and you can easily separate different orders. And then you basically, as you go, you learn different orders. So this is the main computational idea. The problem with this idea, and again, I'm giving some thoughts. I'm not digging deep in every single subject, but the problem with this idea is the following: that do you know how Amazon warehouse works? Basically, you position different shelves, very different objects. So you don't put, and you can have a random network. Here you have CD player next to the T-shirt, and the next part you have, I don't know, the electrical kettle. So when you pick up from the shelf, you never make a mistake because it's very easy to discriminate T-shirt from the CD player. But if you position close by large and small T-shirt, it's very easy to make mistakes. So the customer can get the mixed up order. That prevents you very random specific errors, where it allows you to discriminate between different objects very, very easily. And that is based on the fact that you can keep in mind all this hash table. You can keep all this information in. You can remember this. You go to shelf number 72. You pick the T-shirt and send it to the customer. And it's a good idea because it allows us to discriminate multiple orders that create a huge space. It is a very digital and very, very easy to deal with. The problem with that we are not like that. When I ask you if lemon and orange, apple and orange are far away, but lemon and orange are closer by, you need to have internal representation of these distances. And you need to have some way to compute them. So one of the big problems in this field is how to, given this random projection, how to still preserve the distances and how to make a generalization and not kind of, and combine a very precise discrimination between orders with a generalization property that one order is more similar to another, and to have a map of orders in the same way like we have a map of colors. This is a very open question. I will tell, if we have time, I will take a little bit about the recent work, again, from the same lab of Bob Data, who actually find some structure of the order responses in the cortex, but that given if I have time. What's going on with the time now so far? I have about 20 minutes, right?
Yeah, about 20 minutes.
Okay, so I more or less finished now talking about general organization of the olfactory system, and I would like to talk a little bit about the main processing. Any questions?
I think in the chat, people can raise their hand. They have something.
Thank you for posting the link to Gordon Shepherd. Thank you, Sharad. I don't see any questions, so let's move on. Okay, so let's talk a little bit about the processing, and we come back to the same picture, and we can image glomeruli. We can image activity of the glomerulus, and we can steer on the movies how the different receptor, different glomeruli got activated by the order, and we can formalize this activity. We can basically put them on a time and glomerular level, and we basically can put this kind of patterns for each order or mixture, and we can study forever. The problem with this are all the signals are the same. All the features of the signals are equally important. For example, if I eliminate this glomeruli, this signal, can I still recognize butanol? Or if I shift the glomerulus together, at this important timing or not? What features of this code is relevant for the processing of information? What's interesting here, that we can measure the correlation with the stimulus. And here they work in 2006 by the first authorized sports. They measured the activity, and you see that this is a movie as it progresses along this axis of time, and you can measure the time course of individual glomeruli. This is actually sniff modulated signals, and you see that some glomeruli activated earlier, some glomeruli activated later, and the picture actually evolving in time. How important this temporal information, and how this is kind of what brain is actually reading. This is a subject of the second lecture. Currently, what we can see is the following. For example, if you are an electrophysiologist, I ask you, can you detect the signal of the concentration of the brain? And you measure the signal. At a low concentration, you activate one glomerulus, then you activate two glomerulus, and you activate many glomerulus, and you can read the signals of a concentration by counting number of active glomeruli. In addition, you can look at the one glomerulus, and you can see that glomerulus number one increases amplitude as you increase concentration. So as a physiologist, you put electrode here, or you put your microscope here, and you can read the concentration of the outside stimulus, and you can find a very nice neural correlate. You can also find neural correlate like timing, because high concentration evoke glomeruli earlier than weak concentration. So there's a many ways to read the code. The question that I would like to ask you, what features of this code are actually relevant for the perception of the order of the concentration? That is actually not answered questions. What we know many correlates, but we don't know what is actually read and how they contribute to the perception. Activation of the glomerulus are different in time. So this is the work in 2009 by actually Vachovia group, the first of this Kerry. This is they measure the activation of the glomerulus to the sniff cycle. This is exhalation, then mouth stop inhaling. That's where the odors start getting to the nose. And they measure the latency of activation of the glomeruli, and some of the glomeruli activated early, and some of the glomeruli activated 300 milliseconds later. So this is the cumulative distribution of the glomerular activation. So are all of them relevant? This is a very important question. When actually animal make a decision, all glomerulus, we're talking about combinatorial cord, do all of them contribute equally to the combinatorial cord? And I will talk about it I would be able to talk in the next 15 minutes. Now, what the property of the next level on the mitral cells? Now we can image them, but before we image them, we can put electrode and measure, present multiple orders and record spikes. And here's an interesting, again, historical experts. In 1950, Lord Adrian, who got the Nobel Prize for discovering action potential, was very interested in olfaction. And he made a crucial experiment. He actually published multiple papers. He's one of the first who pay attention to the oscillation in the olfactory bulb that I'm not going to talk about it. But this paper was highly cited as a paper describing oscillation, but not many people read this paper till figure 9. In figure 9, he did a very interesting experiment. He put electrode in the mitral cells of the rabbit, in an anesthetized rabbit, presented the stimulus, this is stimulus presentation, and measured the activity of the neurons. Okay, this is, and what he writes in the figure caption. In a very deep anesthesia, when bulb is quiet, a moderate olfactory stimulus sets up a discharge of impulses in mitral axons at each inspiration, and there are no impulses between. Figure 9a. In less deep anesthesia, the olfactory discharges olfactory discharge appears against a background of continuous irregular activity, and as the anesthesia lightens, the continuous activity becomes more and more prominent until it may be no longer possible to detect any changes due to the stimulus. Figure 9c. Look at this. This is continuously firing cell. You don't see any response. What's going on here? The interesting part that for many years, people ignored this finding. People did not read this paper till figure 9, and they were studying the mitral cells activity in the anesthetized state. And this is a typical response of the mitral cells in an anesthetized state for two orders. Very strong response to citral, or to amyloacetate. This is strong response to amyloacetate, and almost no response to citral. Exactly the same mitral cell, just the same mitral cell, was recorded in the awake mouse when poke the nose into odor port, and there's no signal. We lost the signal, completely lost the signal. So what's going on? Where the signal disappears? How it's actually going on? How the signal is actually is processed? Because we're interested in awake state. We're not interested in the processing of information in awake state. So let's look at the activity. So if you record activity of the mitral cells, one mitral cells in response to odor, and this is a raster plot. This is approximately 70 trials. You can average, you see this, the signals, odor presented here, a little bit excitation, inhibition later, very weak responses. We can study this, but it's boring. What's happening that mouse is sniffing during the same time. And the inhalation are not aligned to the sniff. So the first thing that we did actually, we align all the sniffs to the inhalation and immediately got a very profound response. Then becoming very obvious that response are very strongly locked to inhalation. When I tell this story, I'm a little bit embarrassed that it hasn't been discovered before. Actually, it has been discovered almost simultaneously in two work of now Uchida from Harvard and us, that is, if you look to the inhalation, you get a very strong responses. What was more interesting is that you may notice that different trials, mouse have a different duration of inhalation. This is longer, this is shorter. So what we did furthermore, we stretch and compress. We did temporal warping to make them all equal distances. And we deplotted the spikes in the same plot, and we saw that a histogram became even sharper, which means that the spikes were locked not just to the timing from the onset of inhalation, but to the phase of the sniffing cycle. And they probably carry a lot of information. When spikes are precisely locked to some events in the neural system, they may carry much more information that is just firing rates. So the conclusion of this work was that the mitral cells activity locked to the phase of the sniffing cycle. They're very precise. The precision of this activity is about 10 milliseconds. Now I would like to pause here for a second. So the precision of the signal of the spikes going from LGN to the cortex on the visual processing, also about 10 milliseconds. The retina is actually much more precise. It's about a few milliseconds. But that's the signal that goes to the brain, about 10 milliseconds. The temporal scale of the visual scene is about 100 milliseconds. If you watch the movie, the temporal correlation is about 100 milliseconds. And the precision of the spikes is about 10 milliseconds. And this timing is actually similar to olfaction, because inhalation, the time when the olfactory scene evolved in the nose, is about 100 milliseconds, and the precision about 10 milliseconds. Maybe this is a much general property of the olfactory, of the sensory processing. So they're precise and they're very, very temporally diverse. So different cells are activated at different time and with different properties. Let me a little bit discuss the subject. By the way, at the same time, there was a very similar work published by a group from Harvard, by now Uchida and Kevin. They saw very similar effect, but did slightly different conclusions. But let's look at the activity of different mitral cells. Sorry, just a second. So on the right side here, you see one mitral cells responding to different orders, and they all lock to the sniffing cycle. So if you notice that, first of all, mitral cell lock to the sniffing cycle, and when order comes, for example, when amyl acetate comes, it's a very sharp response and decay. When the amyl acetate and hexanic acid evoke very similar responses, benzaldehyde actually evoked much early and prolonged response, and methyl salicylate inhibit the cell. So we have a lot of diversity of the responses. This many cells, one order. Some, the same order, sometimes evoke excitatory response, sometimes evoke inhibitory and then excitatory response, and on and on and on and on. How can we kind of manage all this information? Let's look. We have 460 cell order pairs, and we measured that 1/3 of them was initially excitatory and a third of them was initially inhibitory. And here's an example. So the gray line shows the mitral cell response without order, but locked to the sniffing cycle. And you see that when order comes, there is strong deviation from the baseline response. And we plotted for multiple cells across the horizontal line. This is time. This is exactly the same time in the sniff coordinates. So basically, all of them was wrapped to the same sniff duration, 358 milliseconds on average, and they were ordered in the latency of the response. So this is early responders, later. So hold on a second. So this data consists of we measure 460 cell order pairs, and a third of them was excited responses. Now, let's assume that this is general distribution of the responses for an order. What that means that when an order comes, an order, just a generic order, and we think that we measure just random cells and random orders, then, and we have responses of 50,000 mitral cells, then 15,000, third of them will be excited responses. So I'll change this axis. Not 136, but 15,000 cells. This is a distribution of the order responses of the mitral cells to an order in each individual inhalation. That is information that the cortex gets, and every sniff. This is a lot of information. Some cells are fired early, some cells are fired later. There's a lot of synchrony and on and on and on. What's going on? Cortex doesn't get it instantaneously. You don't get a snapshot of all 350 milliseconds. First, you get this portion of the cell that are synchronized. There's a few hundred cells that synchronize here. Then other few hundred cells are synchronized here. And another group of cells are firing here. So you get a barrage of synchronous activity of different mitral cells that goes to the cortex, and cortex makes sense of it. Why do I choose 10 milliseconds? Because, I mean, that's the kind of serial sequence of events that happening going to the cortex, how we read the information about the order. Why do I choose 10 milliseconds? 10 milliseconds we choose because the average integration time of pyramidal neuron is about 10 milliseconds. It's exact 10 milliseconds, also precision of the activity in the log to the sniffing cycle. So the synchrony at the level of 10 milliseconds probably plays important role. Is it only for mammals? No. For insects, it's more or less a similar ideas. And there was series of work how I'm doing with time. How much time do I have? Vijay, I need to converge now.
You have about 3 minutes, but I mean you can take some more time if you want and stop at a natural point.
So basically, I just I basically can stop here and talk about the significance of this finding on the next lecture. But the point that I'm trying to make that is the information that going from the olfactory bulb to the cortex, and the cortex mind this and do some decision based on this information. The question that I wanted to pose is, are all this activity is relevant? Do cortex or high brain area pays the same attention to these cells, and that cells? And the point is the following: when you do look at the visual scene and you do recognize, let's say, my face, you get a lot of activity from the retina. But I promise you that 99% of the spikes are completely lost, and only a small portion of the spikes that probably tell you about, let's say, shape of my eyes, and you remember maybe now the glasses if you see me in the screen, or my beard. That's what's really important. The rest will be completely lost. So what portion of these spikes are important or not? And that's actually would be the subject of the discussion of the next lecture. Because what I'm trying to make here, that's a very interesting approach to the coding, we found a lot of correlation. We can study the correlation between mitral cells activity and the concentration, mitral cells activity and the order identity, how mitral cells respond to the different mixture, on and on and on. The question that I would like to ask, what feature of this code is actually read by the behavior and actually important for making judgment? And on this note, I will take questions and we'll talk about that processing tomorrow, because I was slower than I was expected. But tomorrow will be also discussion of the experiment that will answer some of these questions.
Thank you, Dima. Questions?
Yep. Questions, guys?
No raised hands.
I assume that everything was very clear. By the way, I will be also reading Slack, and if you want to post the question on Slack.
Offline. And ask
Offline, if you have a specific question for me to address before tomorrow, but try to do it on your today, because when I wake up, I would have much time to change anything. So just think about it in the next couple of hours before going to sleep. But I will be very happy to address some of the questions in tomorrow's lectures.
There's one question.
Yeah.
Priyant, go ahead.
Yeah, another curious question, Professor. So, were you able to reason out why this process seems to have a sigmoidal-like response at the cortex? Like, were you able to reason that out? Like this plot looks like a sigmoidal plot.
I know. I don't know. Honestly speaking, good question. Why it's has a sigmoidal shape, I don't know. But it's actually, if you think about it, it's actually this shape is, if you look at this shape, give me a second. Oops, how do I hold a second. I'm trying to, oh yeah. This shape is similar to that shape.
Oh, okay.
This shape is activation of the glomeruli. Okay, so this is a very similar shape. Now, talking about, oops, sorry, I'm messing up with my, sorry guys. How can I do this? Yep, here. This shape is very similar to the glomerular activation. We can speculate where this is coming from, but basically what's going on that actually odors appears in the nose. So there's multiple factors that may contribute to the latency of the response of the receptors. One of them are basic physics of the odor delivery, because if you think about it, a very simple model, you have a pipe and you have a receptor sitting here. And when you start inhaling odor, odor goes here. And during moving along this pipe, and you saw this pipe is very complicated. Actually, some of the odor diffuse here, and you also have a profile. So here, and some going further down, and the concentration in this receptor will increase gradually. It's not a step function. It's not like an image in the retina appears instantaneously. Now, some receptors are very sensitive, will respond, will respond earlier, and some that are less sensitive will respond when the concentration reach a higher level. That is a basic biophysics of receptor activity. Now, then the signals goes to the mitral cells, and mitral cells, or to the receptor axon, to the mitral cells, and mitral cells got activated, but also they got also inhibited, and there is a lot of timing, shift of timing. What sets up that distribution? A very good question. I don't know.
But I think the explanation that you gave kind of connects the dots, Professor. So I think I'll also try to understand, try to reason out what could be it, and maybe mail it to you or send you a message.
Yeah.
Thank you.
Okay. Other questions, comments?
I had one question. Is this plot very different for crucial odors? So say urine of a fox or something that you're afraid of. Do you expect to see or for mating, right? I mean, do you expect to see only one receptor and everybody else doesn't respond? I mean, you just want to have no yes/no signal.
Actually, I would be talking specifically about that in the next lecture, and I'll present the so-called primacy coding model that only the very early receptors are important, but activation of the rest of the receptor there's almost no order that as you increase concentration evoke small number of receptors. As you increase concentration, the receptors are promiscuous, they start being activated. But some receptors are super sensitive. So maybe the receptors that are sensitive to the phosphorin, they will trigger here somewhere early and trigger the behavior, and you may ignore the rest. But the distribution of that plot most probably defined not by the order significance or order relevance, but by physics of the receptors and mitral cell processing. The rest is super interesting because what I will try to make a case on the next lecture, that now we develop a tool to judge importance of that signal versus that signal, and probably when mouse sense a danger, they respond very early. I did not ask Sharad to ask this question, but it's a nice segue to the next lecture.
All right. There don't seem to be any more questions.
During
We got a good chat as well.
during your experience, do you release the odor in the pressurized manner, or it reaches the mouth through simple diffusion? Just a second. So odor delivery is super important because it's kind of touching very important subject. In the majority of the cases in olfaction, people were studying the odor responses by taking a Q-tip and bringing it to the mouse nose and seeing what the mouse is doing. This is the level of the experiment that had been done before. And that is very different from the level of experiment that has been done in vision. When you set up your subject, you control the eye movement, you present very well calibrated display, and you know exactly what's going on the retina, and you also track the eye movement. We do more or less the same with olfaction. We calibrate the stimulus, we flow the odor by mouse nose, and we trigger the odor valve, and we know exactly when the odor hits the nose using so-called photoionization detector. And we calibrate it relative to sniff cycle. So like checking the eye movement, we checking the sniffing, inhalation, and exhalation. And that the result of finding the high temporal precision is due to very well controlled stimulus. So to answer directly this question, the air always flowing by the nose, and you insert the odor into the flow. So we try to avoid any pressure signals in the line when we deliver odor, even though it probably cannot be avoided completely.
Okay, no more questions. Thank you. Okay. All right, let's thank Dima for a nice talk. And so that concludes the session for today. We'll come back tomorrow for the second talk by Antonio and also the second talk by Dima. Thank you, Dima.
All the best. Have a good day, and ask me a question during Slack. But today, tomorrow I would have much time or little time to prepare on my time.
All right.
Okay.
Take care, everyone. We'll meet up tomorrow.
Yeah. Bye.